Okay, welcome back, AP Biology. Today we're going to talk about nucleic acids. I do expect that you remember some of this information from ninth grade. So let's start out by talking about the monomers of nucleic acids, which are nucleotides. So nucleotides, you might remember, are guanine, thymine, adenine, cytosine, and uracil from when we talked about DNA and RNA. But there's also a few others that we've mentioned and maybe didn't point out that they were nucleotides. For example, ATP, adenosine triphosphate. Notice the adenine in that. So there's 3 phosphates, a sugar, and a nitrogen base, and that's what makes up a nucleotide. GTP is also an example of a nucleotide. NAD plus and FAD we talked about with cellular respiration. And then cyclic AMP will be a new one for us. We'll talk about this with signal transduction. It's a way of getting a signal amplified from outside of the cell to the inside of the cell. So that's yet to come, second semester. So nucleotides, what do they do? If you think about DNA and RNA, you know that they store information and they can transfer that information from point A to point B. They also serve as chemical energy. So we're very familiar with ATP being the energy currency of the cell. There's also guanine phosphate. So using a different nucleotide or a different nitrogen base, we have another energy molecule. And then I was referring to the cell signaling that happens with cyclic AMP. And then we've also talked about cofactors. For example, we used coenzyme A last biology class in ninth grade, and we've used FAD and NADP also. So with cellular respiration, we talked about these coenzymes, and a coenzyme is something that's going to help a substrate fit better into an enzyme, so they cooperate. So here's the general structure of a nucleotide. All nucleotides have a phosphate, a sugar, and a nitrogen base. That sugar is a pentose. It's a 5-carbon sugar. It's either ribose or deoxyribose. Can you guess which one this is? On the next slide, I'm going to talk about how to tell the difference between deoxyribose and ribose. But basically, I know right away that one is ribose because of this hydroxyl on the second carbon. So the nitrogen base, you want to know, is attached to the first carbon, and the phosphate is attached to the 5th carbon. So I've seen that as a test question before, so look out for it. So here's your deoxyribose versus your ribose. Notice in the 2' carbon, you have a hydroxyl group or you have a hydrogen group. So it makes sense. Deoxy, you've taken the oxygen away. So this oxygen right here from the hydroxyl has been removed, and that's the difference between the ribose and the deoxyribose sugar. So there's 2 different types of nucleotides. Notice one is a double ring, one is a single ring. Which is which? We need to know. So the purines are the double rings. They're the smaller name, but the bigger molecule. That's one of the ways I remembered it. So you see adenine and guanine, and I don't know if you remember your chemical symbols, but Ag is silver. So my mnemonic device is to say pure silver. So a purine and silver is the Ag portion. The pyrimidine is the smaller molecule, but the much bigger word. I know that's a simple mnemonic, but that's what I use. And then cytosine and thymine both have Ys, as does pyrimidine. And then we know uracil replaces thymine, so if thymine is a pyrimidine, then uracil must be also. So later, second semester, we'll talk about DNA, and we'll find out that there's always a purine bonding with a pyrimidine. So here's another bond. We've talked about glycosidic linkage, ester bonds, peptide bonds, and now we're talking about phosphodiester bonds. So the phosphodiester bond occurs between the 1, 2, 3, the 3' carbon, and the phosphate that is attached to the 5th carbon. So we have to bond from the 5' to the 3' carbon there. Basically, it's the phosphate. So that's the phosphodiester bond between the hydroxyl group. So this causes DNA to have directionality. It can only run in one direction because that phosphate group can only bind to the 3' carbon. So new nucleotides, when forming the polymer, when going through dehydration synthesis, will only be added at the 3' end. So I like to say add free to the 3. So a free nucleotide is added to the 3' carbon. So that is that creates that directionality. So you can only add, so I'm headed in the 3' direction. This is your phosphodiester bond between the phosphate and the hydroxyl. And then we're going to continue in that direction, adding one nucleotide at a time. So the enzymes that we use to form DNA and RNA can only fit that the 3' and the phosphate together. So that's what creates that directionality. So I think we have a pretty good concept that DNA and RNA serve as blueprints. So they're going to make exact copies of DNA minus, mutations that occur. But the goal is to make exact copies of the DNA. And then each cell then will make the proteins that it needs by transcribing that information into RNA, and then RNA will be translated at the ribosome to create proteins. So we're making lots of connections here. So every one of your cells should have the same information because they all go through that process, as well as your gametes creating new cells that are going to become future generations should have the same information as well because they're following the same process. So for each of the macromolecules we're talking about, we want to understand the structure and the function. So the function of nucleic acids, referring to DNA and RNA, has to do with genetic information. So either storing it or transferring it from point A to point B. So for example, from the nucleus to the ribosome. So RNA is ribonucleic acid. It is a single helix, so one strand. And deoxyribose DNA is a double helix, so it has 2 strands, and those 2 strands have a relationship and that they're complementary, which means one side determines the other side. So this is RNA over here, just a single chain, and this is DNA over here with a double chain. So a little bit more about DNA. So DNA, we said, is a double helix, so 2 pieces twisted around each other. They're formed with phosphodiester bonds. So in between 2 nucleotides, in the growing chain, you have phosphodiester bonds. In between the 2 chains, so to be clear, you have hydrogen bonds. So between cytosine and guanine, you have a triple bond, and between adenine and thymine, you have a double bond. So if these weren't labeled, I would still be able to guess which nucleotide they were, because seeing the triple bond right here, first of all, narrows it down to cytosine or guanine. Then I look at the double versus the single ring, and I know that guanine is a double ring because it's a purine, and cytosine is a single ring because it is a pyrimidine. So that would allow me to identify which is which from a diagram. So if you had to do that, that's a skill you could use. When you see this double bond here, you know that you are limited to thymine or adenine. Since this is a double ring, I'm not using uracil, or a double helix, I'm not using uracil. So the double bond between these 2 limits it to A or T, and then again, the pyrimidines have a single ring, so that makes this thymine, and the purines have a double ring, so that makes that adenine. So hydrogen bonds are really important because if you remember, they're just attractive forces. They're van der Waals forces. They can easily be broken. If I need to make an exact copy of this, I need to be able to access the information that's inside. So hydrogen bonds can easily be broken, and that allows me to bond new pieces in order to make the copy. So that allows it to serve as a template. So one of the other nucleotides we talk about is adenine triphosphate. So adenine 1,2,3 triphosphate. This last bond, because of its position, has a great deal of energy stored in it. So it's typically thought of as the energy currency of the cell. I don't know if you remember, but this is made in the mitochondria, correct? So all of our cells have mitochondria that will make energy. So you can break off 1 bond, 2 bonds, 3 bonds. You could have adenosine triphosphate, diphosphate, or monophosphate. The more bonds you have, the more energy there is. So energy is stored in the bonds. CAMP is a new one for you, cyclic AMP. We are going to get a chance to talk about second semester when we talk about signal transduction. So a molecule will bind to the outside of a cell. On the membrane, you have proteins that are receivers. And so some messenger, some signal, like maybe a hormone will bind to that receptor protein. And then that information has to get its way inside of the cell, telling the cell what to do. Eventually, you're probably making some sort of proteins, so you have to get to the nucleus to send that signal to start the protein synthesis. And that is kind of the job of cyclic AMP. It's going to help amplify that signal and pass it inward. So it's a secondary messenger in many of our pathways. One example down here, the regulation of glycogen and sugar, as well as lipid metabolism. So we'll use the example with insulin and glycogen in second semester, and then this will make more sense. So NAD plus nicotinamide adenine dinucleotide is just a fun word to say. That's a coenzyme that's found in every cell. We know it simply as an electron carrier. So this, because it is charged right now, it can accept an electron, right? So we're going to bind with a hydrogen, in which case it's accepting an electron, and then it transfers that from one chemical reaction to another, or from one part of the cell to another. So this participates in redox reactions, reduction, oxidation reactions we'll talk about when we do photosynthesis and cellular respiration. And then we also have flavin adenine dinucleotide. So that adenine kind of gives you an idea that it's a nucleotide, right? Other than the fact that its name is right there. And it's the same sort of thing. It's also an electron carrier. It can bind a hydrogen, in which case it's going to carry that electron from one chemical reaction to another. And so that one we are a little bit familiar with already. So that's my summary of nucleic acids, short and sweet. Most of it you knew before. Maybe the FAD and NAD and cyclic AMP were not as familiar, and so that was new. The phosphodiester bonds should definitely feel new to you. But hopefully that explains the directionality and why we grow DNA from a 5' to a 3' end. So that's all I have for you today. I hope that was helpful.